Okay, so we are recording now. Welcome back to the first afternoon session of this workshop. It's really a pleasure. It's just getting better and better. We now have Peter Dittrich, who is the founder of the ideas around COT. So a very important inspiration for our work. And we are all very happy to have you here, really. It's a big moment to gather these people. Like, we have been waiting for 10 years to do this. So now it's a big moment. Okay, go ahead.
Yes, thank you very much, and thank you very much for the invitation and the funding, of course. And in this talk, I will not talk about organizations, but maybe about a bit other topic about goal-directedness and about maybe semiosis in goal-directed systems. So it will be very highly speculative talk. Yeah, I would like to give you some strange thoughts, and maybe there's something. Yeah? But this, maybe you should understand the talk like this. So the talk will be about the idea, maybe there's semiosis in goal-directed systems, and maybe how to recognize this kind of thing. So the aim, okay, here's again the aim of the talk. So I would like to think about the role of semiosis. Semiosis means that signs are used, so some meaningful signs are used in goal-directness or goal-directed systems. And when we look at those kind of systems, many goal-directed systems seem to be semiotic systems as well. And by semiotic system, I mean any system that is using a sign. And for example, living systems are a special case. So can we make this view more precise? So for example, can we use some kind of formal approach or some kind of measurable approach to that? And there's a little sub-hypothesis that I would like to present you is that if you look at a feedback control system like that you have in control theory called Regelkreis in German, that is essentially also a semiotic system. And all that I know, all of them are semiotic systems. And we will see that they employ a so-called contingent mapping. And that's what I want to use to make this more precise. So the talk goes like that. First, I present you some wild speculative thoughts. Yeah? And then I try, in the second step, I try to operationalize them with some kind of formal theory and computer algorithms. Then I would like to show you some preliminary tests on some data, and then try to align these thoughts again with goal-directedness to come back. So the basic speculative thought here is that I would like to suggest or try to suggest a new physical property. So by that, I mean a property that can be measured by terms of scientific physical experiments. And this new physical property characterizes physical systems with respect to their ability to implement contingent mappings. What do I mean by a mapping? I mean here the mathematical concept of a mapping. Namely, a mapping relates to set with each other. And examples for such mappings are the Morse code, for example, or the genetic code. Genetic code is a mapping between triplets and amino acids. And by contingent, I mean that the mapping on the same sets can be different. So it's possible to implement an alternative mapping with the same system, but that maps the sets in a different way. In the Morse code, it's obvious. In the genetic code, in a way, also, we have examples for that. So this contingency is a very important property for mapping, because otherwise there are many mappings in the physical world. And what do I mean by system? Of course, what is that? So I mean things like the atmosphere chemistry of Mars, or the combustion chemistry of methane, or the machinery of mRNA protein translation, or maybe also a system made of a bunch of transistors and resistors or a silicon wafer. All these are systems, let's say, that we can now study with respect to the ability to implement so-called these contingent mappings. Okay, so contingent mapping is also called, as you have seen there, Morse code, genetic code. People also call these contingent mappings codes in general. And in the context of biological systems, there's the notion of organic code that has been coined in the domain of biosemiotics, for example, by Marcello Barbieri. And we have developed a formal concept that is called the molecular code, which is then a contingent mapping made by molecules in the domain of chemistry, of course, as you know, that we really like these things. So what is now, because the ability to implement a contingent mapping differs in different physical systems, we can now use this kind of thing, if it really works, to distinguish physical systems. So there are some physical systems that can do it and some that cannot do it very well. And interestingly, we find the property of being able to implement contingent mappings more often in living systems, maybe in every living systems, and also in information processing system and communication systems. And so it seems that this property could be necessary for semiosis, for the usage of science. So it might be a necessary property. So if you don't have the property, maybe it's not possible to make meaningful sign-mediated communication. It seems to be necessary for life. It could be maybe a necessary condition for life, and also to make an information processing system in the sense, of course, as computer science is using the term information processing, not in the sense that maybe Shannon theory is using the term, okay? And so now let's go to the more operationalizing way. So now we want to put this into more concrete terms, and we have done it in two domains. One is the reaction network domain, using reaction networks, and one is cellular automata domain. And both domains can be used to model the real world, physics. You can make physical models of the real world, models with both type of approaches. It has been shown maybe to the cellular automata, are famous, maybe a study work by Stephen Wolfram, who used it as a model of the world. And reaction networks, of course, we have seen can be also used for that. So basically both approaches now serve as a way to represent the world, and that can be made more and more precise by physical experiments. So you can take them as models of the physical world. So now I will start, of course, with reaction networks, and I will focus here more on the reaction network approach, but I will also very briefly show you with the cellular automata. So now going to the chemical level, how can we make this more precise? So I will just show you the meat, yeah? Okay, this is just, okay. It's the same again, just making on the level of molecules. Now we have to, of course, move this thing to the molecules. So we have to say now, what is a mapping? So a mapping in the simplest case is to map molecules to molecules. And what is the simplest thing we can think about? Two molecules are mapped to two molecules. So that is the simplest way that we can think about. And now we have to, in a way, find ways how we can make mappings with molecules. And I will show you that now, maybe using an example. That's the easiest way to do that. So now what we now want, okay, so there's also, I give you some kind of background here where this is coming from, these ideas from the domain of biosemiosis. Also Jacques Monod, maybe the book Chance and Necessity has an interesting chapter inside that is already discussing this thing, that the mappings in biological systems must have some kind of contingency, and it's continued also in this biosemiotics domain. And a very nice paper by Marcello Barbieri. Okay, and my personal motivation was also maybe to distinguish flame and living organism, which is not that easy to distinguish, and maybe to distinguish science and molecules that are not science. How can we distinguish semiochemicals from metabolites? And it's maybe it should also contribute to that. So let's go. What we want to do is now we want to find a mapping. Okay, so we want to we want to define this contingent mapping now on the level of molecules. And we call this contingent mapping on the level of molecules, we call a molecular code. And what is a molecular code? It's always with respect to some kind of chemistry we are looking at. So with respect to some kind of chemistry, a molecular code is, first, it has to be a mapping between molecules that can be implemented or realized with other molecules. So I can use a bunch of molecules, and these molecules I can use to implement the mapping between the other molecules. Okay, that's the first thing. But the second property is that it must be contingent. That means that it must be possible to change the relation between the molecules. So to change the mapping of the same domain and codomain by just exchanging the molecules that are implementing the mapping. And I will now show you how that works in an example. Okay, so I have now to switch the slides. I'm sorry. I have to switch to the other slides because they don't work. Okay, 1 second, please. Computer, no share.
I don't need to change.
I don't have to change. Yeah, you're right. Yes. Yeah. No. Okay, now it works. So now you should see it. So here's now the example of the reaction network. So in this network, we have A1 is, let's say, a substrate. E1 is an enzyme. And the E1 is now converting the A1 to a B1. And you see the reaction rule is simply A1 plus E1 reacts to form B1 plus E1, yeah, just a rule. And you see it here in the graphical representation. And here now you see the full network. So we have more rules like A1 can also be converted by the enzyme E3 to B2, and A2 can be converted by E4 to B1, and A2 can be converted by E2 to B2. Yeah. So that's a reaction network. The names of the molecules are of course arbitrary, but I put the names so that we already see what the structure is, okay? And draw it also that you can see what the structure is. Now I want first to explain what does it mean to realize a mapping with molecules. And then second, I will explain what contingent means. So realizing a mapping, you first have to choose what you want to map from where to where. And here you see now okay, here it is red or the orange part. These are the molecules I want to map now to the blue molecule. So this is a set, so the red set and the blue set, and I want to map the red set to the blue set. And how do I do it? I have to find a context. I call it a context, a set of molecules. And these molecules are used now as a machine to implement the mapping. And here, of course, you see, I have to use these green molecules. These two molecules I have to use. And this is now my machine to implement the mapping. And how do I implement it? Yeah, I just throw into these molecules, I throw in, let's say, the molecule A1, and I call it a sign. So don't, I mean, now you can kill me for using this term, yeah, but please, I just use it as a term because it's coming from some historical thing. So I call this red set, I call the signs. And so the A1 now is thrown inside, and then I let them react, yeah. I create the closure, so and then you see what everything that is produced, that is a closure, that is computed, and you see now the B1 is now inside, and the B2 is not inside. So the requirement is now this molecule. I mean, there should be a molecule from the set inside, but not the other one, yeah, because otherwise I cannot decide what has been computed. So now you see this thing is mapping the A1 to the B1, and of course, when I throw in the A2. Then I get, I mean, obviously the B2 as a product. So now with this kind of chemistry, I can compute this mapping, okay? So now the question is now, is this mapping contingent? And that means now I can implement an alternative mapping on the same domain and codomain. So let's try, okay, you see it already. When I take now this molecules as a context, so I can now do the same computation, then you see that I can now map the A1 to the B2 and the A2 to the B1. Yeah. So that means now that this mapping is contingent because I can implement an alternative mapping on the same sets of molecules. Okay, just by choosing a different context. Okay, so I can do that. Yeah, okay, so here's again illustrated. Yeah. And so now we have these two different mappings. So now you might ask, okay, so that is easier. So maybe we have these contingent mappings everywhere. And I show you now another mapping here. So you see now again two sets, E1, E3 and B1, B2. And again, we can implement a mapping between these two sets. And you see this now with the pink arrows. Yeah. So I can map the E1 to the B1 and the E3 to the B2 by using A1 as a context. So when I have A1 in my vessel or so I can throw in E1, I get B1. I can throw in E3, I get B2. So I can compute this mapping. But this mapping is not contingent. So it's not possible to change the mapping, no matter what you do. So you can do whatever you want in this physical world. It's impossible to change the mapping. So in other words, in this kind of world, if I give you the red and the blue, and I tell you there's a mapping between, you can deduce the mapping from the physical laws of that world. So the mapping is here determined by the physical laws of the world. There's no way to generate any contingency between these mappings. Whereas in this other example, when I give you now the red and the blue set and all the physical laws of this world, you cannot relate these two sets to each other, the mapping. Yeah. So maybe the interesting property now is that the property of the mapping to be contingent or not cannot be derived from an actual implementation of the mapping. So when you see the mapping operating in an organism, you don't know whether it's contingent or not. You have to know the alternative worlds, and the alternative worlds depend on the physical laws. So it's not some magic or so. It's just the physical laws that determine the alternative settings. I mean, let's see. Okay, so let's do, so that's now the technique. And we have now made a tool that, given some kind of network like this, it finds all the molecular codes. So it finds all the contingent mappings. Okay, so and then you can apply this to networks. Yeah, and we use then an, okay, I'm again sorry for the strong term, we call it semantic capacity. So the more codes a network can realize, the more semantic capacity it has. Yeah, because our impression is that they help to make communication. So we call the semantic capacity simply the number of molecular codes a network can implement. Yeah. Okay. So now we can measure it. So we can now search for these things. And obviously, when you look at the translation mechanism of a cell, so where you have the mapping from the from the nucleic acid triplets to the amino acids. So this is a genetic code. Essentially, it is implemented. The context here is the tRNA and the loader enzymes and so on. That's the context. And of course, you can change the genetic code. There are examples even in the biotic world where the code is slightly changed, and you can also change it by experiments. When you make a reaction network model of this kind of chemistry, including all the possible loader enzymes, of course, you get a huge combinatorial number of possible mappings and codes. That is in a way obvious, I think. So depending on how you model that, you get millions of codes. That Dennis, for example, here 1.5 million codes, like Dennis Görlich has computed here on this kind of network. I will not go into detail because it's quite clear, but you also have these contingent mappings at every other levels of the cell. So you also have it here on the level of gene regulation. So in gene regulation, what you see here the DNA. So this here is a, okay, so this is a DNA here. This is a DNA. It's also a molecule, yeah? So DNA. And on this DNA, you have regions that are called genes. And these regions, they have a coding region that code for the protein. And so this is a coding region that codes for code protein, the green thing. And then you have also a region that is called the promoter region that determines which other protein, which is here the red part, is activating this gene to make this protein. And now you see the mapping here is between the red circle, that's the transcription factor, and the green thing. So that is now the mapping. So the mapping is between the red and the green protein here. That is the mapping. And that is also contingent. Why is it contingent? Maybe you know that it's very easy to exchange the coding region here. So we can just take this and create, let's say, the protein of the coronavirus or whatever you want. Yeah. So you can put it here. So you can really, it's very easy for the cell and for a biological scientist to change this mapping. Extremely easy. And maybe just to, one interesting thought is, before, I mean, in the example that I had before, in this example, the triplets here are the signs. Oh, I'm sorry, it's always delayed here. the triplets here are the signs. So this is DNA that is mapped to protein. And in this example now, the DNA is here the context that is implementing the code. Yeah. So you see that these coded contingent mappings are somehow networked with each other. And you also find them in signal transduction networks. Just very briefly, signal transduction networks, you have proteins, and a protein, like a kinase, can activate another protein to become active. Yeah, so you can phosphorylate a protein to become active. So A becomes active, and then the B can do something. And this is not a contingent mapping. It's not contingent because this is physics. This is chemistry here. That the S attaches to the A, it phosphorylates it, and make the B. So this is not a contingent thing. So now you ask, signal transduction, maybe it's contingent, and indeed it is. But it becomes contingent when you make two steps. When you have a cascade, and you know there are many cascades, it's not just a single process in cells. When you make a two-step process, then the mapping between this one and this one becomes contingent. Because in a cell, you have, let's say, does not exist, yeah? But you only have, let's say, this part, and you see now S is activating this, is activating this. So now assume that the cell is removing this because you just kill so switch off this gene and you express this part. Now the S is activating this, which is inhibiting this one. It should be like that. So I cannot think while I talk. So sorry. But you see now it depends on what is expressed in the middle part, whether now the S is activating the D or not activating the D. And that means if you have a two-step process, it's a contingent relation. It would be very speculative. It would very high speculation to say that this is maybe the reason why the pathways are so long, but this I don't dare to even to say. Okay, so we we also looked at metabolic networks, And it's much harder to find the contingent mappings there. Very seldom. And even those that we find are a bit strange. They use, for example, currency metabolites to do the mapping and so on. So we hardly have, I mean, we are no experts in that. So what we can say is we find them it's very hard to find them even in very large networks, but there are some, yeah, but there are some of these contingent mappings, just to show you that. And then we also look at the atmosphere chemistry of Mars, the atmosphere photochemistry of Mars, here a model from a textbook, 31 molecular species, 103 reactions. The reactions look like this, just to get an idea. And you see in these reactions, you have this anorganic species here, and you have also light. H nu here, that is light, that is driving this chemistry out of equilibrium. And this chemistry, it has also nice lattice of organizations, by the way, really beautiful, especially on the light side of Mars, where there's the sun. At the dark side, it's not so nice, as you can imagine. So, so now when you now look at the codes, there's not a single molecular code in the whole network, even if there are many, many closed sets, but there's no way to implement a contingent mapping, even with this kind of chemistry. And so even there are 5,000 closed sets, yeah, but there's no way to make this contingent mapping in this chemistry. We also looked at combustion chemistries. In combustion chemistries, you have, here's an example of methane combustion, 340 reactions, 37 species, ethane, 750 reactions. So these networks are pretty large. So we also studied all possible molecular codes in these chemistries, and there were no. So it was not possible in none of these chemistries to make a contingent mapping. And so maybe now you can, from this, you might somehow be very careful, of course, in saying things, but it seems so that in the chemistries that we find in living systems, it seems to be easier to implement these contingent mappings than in these unorganic chemistries that we have here. Maybe it's an error in the modeling, maybe it's an error in the way we are looking at the system, so we have to be very careful. But these preliminary results seem to be that the semantic capacity is high and here is low, and this might then, from this you might also very carefully say some hypothesis saying that over the course of prebiotic and biotic evolution, the semantic capacity of the chemistry that is incorporated by life tends to increase. And by incorporating by life, I mean making accessible to, if you say life and the organisms are created are now autonomous agents, yeah, that get access to the chemistry of the universe, yeah. And we get more and more access to what the universe offers in the domain of chemistry. And you can then say maybe life gets more and more access to more and more contingent mappings, yeah, increasing that. And you can be very speculative. Okay, I will stop the speculation now. So the approach can be generalized, not only to chemistry, also I think to any kind of media that can be made, that we can model in a way. And I just show you very briefly, just on 1 or 2 slides, how this is done in cellular automaton. So what you see here is a cellular automaton, a one-dimensional cellular automaton, and you see here each cell can be on and off, or there are just 2 states, and it's going from, the time is going from top to bottom, that is time. And you see here only elementary cellular automata. So there are only 256 of them. It's the most basic cellular automaton you can think about. And you can now also measure now here the semantic capacity basically in the same procedure as I did before. So, but you have to define what is now your thing you want to map to where. So what is the mapping? And that is a bit, I mean, you have to now to decide what to take. So you may say, okay, maybe you should do something else. But here, the sign, so what you want to map is the state of this cell. Then you iterate it in a time, and then you take the state of this cell, yeah, which may be somewhere else. Of course, it should not be here. I mean, that would not make sense, yeah. But you take another cell and you say, this is the set I want to map it to. So that means that you have 2 states here, 0 or 1. You have here states 0 and 1. And now I want to map these states to the other. And now the context, okay, now it's blue. I'm sorry, that was green before. So now the context are just the cells that are surrounding it. So I just take the cells that are surrounding this cell, and that is the context. And now I can ask, is it now possible to implement a mapping? Yeah. So for example, this context, you see it's the same context, yeah, the same context. Now this black is mapped to black, and the white is mapped to white. Yeah. Okay, so the mapping works. Now, can I change the context? Yes, in this case it works. I can change the context, and now the white is mapped to black and the black is mapped to white. So in this case, it's a contingent mapping because I can change the context. So now we can ask, is that always possible? No, no, of course not. So it depends on the world. Yeah? It depends on the physical laws, if you like. So there are 256 cellular automata, and you see them here all. So these are all the 256 cellular automata. And here you see the number of codes. So basically the number of possible mappings that they can do. So I measure, how I measure this, I have to, that is more detail. Yeah? But you can now say that the larger the thing here is, the more contingent mappings the cellular automata can implement. And I've also sorted the cellular automaton here according to the complexity classes. So you see here the things that go to 0 all the time, those that go to fixed points, those that have these cycles, some kind of periodic orbits, and the chaotic ones. And you see that the semantic capacity is not so trivially related to the complexity. Yeah, so it does not mean that if you have high complexity here, that you get also high semantic capacity. No, not necessarily. But also having boring automata that go to fixed point might also have a high semantic capacity. They're just boring, yeah, but they can do very well these mapping, these contingent mappings. Yeah, so let's say, so what I want to say here is that there's not a trivial relationship between complexity and semantic capacity. Yeah? And you can study it nicely in these cellular automata. So finally, of course, we need in the future more data and so on, and quantified maybe by information theory, and there's a possibility. But let me now at the end also relate, say to, I want to say something to, so let's see whether it works. So I want to say something about a goal-directedness, and that maybe semiosis may be somehow included or maybe related to goal-directedness. And so first, what we can maybe say is that there are goal-directed systems that use semiosis, yeah, that use science, and they use contingent mappings. So maybe there are also some that don't do it, yeah. But let's say one thing that is doing it is the feedback control loop, and I just want to, now, okay, I'm sorry for the German slide, but so this is a slide showing the typical feedback control loop. So you have the feedback controller here, and you have the system that is controlled here, and you have here something that is measured, and then this measured quantity is then compared with what should be the aim, should be the aim. And actually, this pointed measurement of the goal of the system is actually a semiotic process, because here you have a contingent mapping. At that point, in all of the systems that I know, there's a contingent mapping at that point. And this contingent mapping is moving the state of the system into a semiotic space, into a sign. And this sign then is used in the controller to decide what to do. And when you now look at, okay, this is also in German, so so this is just an example from biology of the different components that you can find in the regulation of the sugar, which we are now experiencing, I suppose, after lunch, how it is done and controlled. And you have all the components, indeed, of this control system you can identify also on the biological system. So that is indeed possible. And also when you, for example, look at these classical feedback control systems, this is also actually a semiotic process where this one is mapping the state somehow, and it's an arbitrary mapping. So whether I map now this behavior of this kind of thing, which is called, okay, it's now, I cannot see it. So I looked up the name.
Of what?
So because things are recorded, okay, I cannot. So it's a centrifugal governor. So you can now, of course, measure the state of your system. The faster it goes, it moves here. And then you map this to opening or closing the valve. And this is a contingent mapping that you apply there. You can also map it in another way around if you want to make an explosion or so. Yeah. So, so it depends. So you have now, so what I would say is now that feedback control systems are essentially semiotic systems. And maybe not. Maybe there are ones that are not. And so let me then conclude in a way. So semantic capacity, at least in this term as I introduced, can be measured and differs in different physical media. And it allows to identify a mechanism of how, or identifying the mechanism of how contingency is realized can shed some additional light on sign-mediated biological information processing communication. And there seems to be a class of goal-directed systems like the feedback controller, at least the examples I showed maybe, that are using these contingent mappings, and they're creating essentially signs, yeah, that have meanings, and to do the regulation process. Thank you very much.
Thank you, Peter. Amazing timing.
Lost it.
And very interesting, like complete. So comments, we have like 10 minutes for questions. Yeah, so go ahead, Francis.
Yes.
Well, the question is, why would it be useful to have a contingent mapping? And the reason I can think of is that the contingent mapping allows more experimentation with different mappings. So some mappings may be more fit than others.
Yes.
Some mappings may be better at suppressing perturbations than others. Like in your example of the governor, the farther away it goes, it means the faster it is spinning. That means the system is going too fast. Therefore, you should reduce the steam. So there you want to have a negative feedback. But in the case of a negative feedback, it's usually so trivial that you don't necessarily need to have this kind of contingent mapping. But if there would be several options of action acting, not either more or less, in which case it would be almost trivial, but let's say you would have 5 different options, then the fact that different mappings that lead to each of the options could be tried out in natural selections, that may be an advantage, gives the system a flexibility. And I think that's what the genetic code does. Because the genetic code has such a liberty in possible mappings, it's interesting to try all different genes.
Yes.
But I mean, you have to explain why the contingency is evolutionary fit, why it's an advantage.
I agree, and that is really a basic question. And that I think there's no definite answer yet. That might be an answer. Another answer is that you want to have a symbol of something. For example, a symbol of poison. So when you want to react to a poison, you don't want to have the poison inside the body. So that means that you want to have a symbol for the poison. It is very unlikely that the poison itself provides you with a symbol directly by physical laws. So that might be also a reason. Also, when you look at the reproduction, like von Neumann self-replicator, you might also say, okay, the mapping allows you to maybe explore alternatives, right? But also the von Neumann replicator is essentially also semiotic in the sense that it cannot replicate its structure. It can only replicate its tape, which is then interpreted to create the structure. So there's the contingent mapping is from the tape to the structure that I'm generating. It is a contingent mapping because you have a universal constructor, and this universal constructor is basically reading the tape, and the tape mapping. So the mapping from the tape to the structure I'm constructing, that is a contingent mapping. That's also in nature a contingent mapping. And so the replication process by von Neumann replicator is essentially using contingent mappings, even if it is not necessary. So when you have, for example, a replicator from Kiedrowski, for example, that is template-based replicating, it's a replication process without the need of contingent mapping. You just read your own structure and make a copy of it. For that, you don't need any semiotic or contingent mapping.
So I might think of another reason why contingent mapping would be useful. If I think about language, language has words which are contingently mapped to meanings. Now, we could in principle imagine some kind of an onomatopoeic language where if you want to refer to a cat, you say meow, and if you want to refer to a dog, you say woof.
Yes.
But that language would, of course, be very limited in the kind of things for which you would find non-contingent mappings.
Yes.
Well, the fact that it is contingent, that dog and hund are completely different words, but they can both refer to dog, allows you to invent symbols for any kind of thing. So you can experiment much more variety of possible things that you express.
So the idea of getting more and more universal, the higher level the symbols become more abstract. Yes. Yeah. Huh?
Can I ask? Can you hear me?
Please. I can understand. Yeah.
Great. Well, first of all, thank you very much for this presentation. It's intriguing, this semiosis idea. It's really sad that I can't be with you in Granada because I have so many questions on this, but just a short one, because of course semiosis is at the heart of the matter of communication design, you know, and so this is really great. What I'm asking myself is, how is the decision happening of the code makers? Where is the decision taking place that they do this mapping and not the other mapping?
Okay, so this is not inside yet the theory. So we just take a code maker and take it. Yeah. So there's no process that is generating the code maker. Yeah, that is not there.
Yeah.
And there's also nothing that maintains the code maker. Of course, the idea is now maybe to join it with autopoiesis, that then we say that only those code makers that can be maintained make sense. And then you might extend it and say, okay, if the alternative code maker cannot be maintained, I mean, you're also off, yeah. But there's currently nothing inside that explains where these code makers are coming from. But that would be an interesting next step, yeah, to think about processes. Yeah,
great.
Of course, very nicely to the project, because it is also the aim, I think, of that project here. And it's an open problem, yeah, so we don't know where the things are coming from. Yeah. Mm-hmm.
Yeah, great. Thank you very much.
Okay, Carl has a question.
I have just one remark that I think you thought of that.
No, please.
This reminds me a little bit of Moore and media automata. These things.
Yeah.
And they exist in vast literature of input-output behavior of Moore and media automata.
Yes.
But the real question I would have is, this is fascinating, and I have been confronted with your ideas for the first time. So please excuse that.
No problem. No.
So may I ask you, what is the difference between contingency and context?
Okay, so the context is the machine.
Yes.
Or let's say really maybe the physical machine that is implementing a particular mapping.
And that particular mapping is due to its contingency, or?
So the contingency is a property of the mapping, or the property of the mapping. Yeah, not of the code maker. So the code maker or the context is just one machinery implementing one mapping. And the mapping is called contingent if there's another context, a different context that implements a different mapping.
Yeah.
But on the same domain and codomain of molecules. Yeah, on the same set of molecules.
I don't understand that, but that's challenging.
Yeah, okay. That is
it should be different. Be a different mapping.
Must be different, let's say non-trivial mapping. So we don't consider constant mappings, for example. It would be too boring. So it must really,
is a clarification. So you propose to define signs as soon as we have multiple mappings, is what you are suggesting. So I was thinking something like.
Of course, I know all the problems.
No. I mean, maybe it's clarification. So I was thinking about a very trivial case. We have the spring of a river and the mouth of the river, and then we have different possibilities. So if we do nothing, there is some path, and then we build a dam. So we have a different path.
Yes.
Would you are saying, implying that then the river has the source is.
So what would be the mapping from where to where?
The different trajectories, the possible trajectory between the starting point and the end point of the river.
So what is the input then? The two states of
the starting point of the water, the spring, where the water comes.
So there are two springs, let's say, A and B.
One spring or two springs.
Yeah, okay.
And then you have the ending point of the river.
Yes.
And then we can imagine different trajectories.
Yeah, and if you can now map this spring to this outflow and this spring to that outflow, yes.
That would be a semantic system.
Yeah. If this would be employed now by some system to maintain itself, maybe.
So then what you require is something else. It's not the mapping. It's a clarification.
No. So okay. So let's say we would now say this system has a capacity to be would have a semantic capacity. So whether it is actually used as a sign processing system, it's a different story. Yeah. So we would say yes, this has the capacity. It has a semantic capacity because you have the ability to implement a contingent mapping.
So
yes.
So actually,
so I would not say that it is a semantic system. So I don't call now the reaction network that we have here, a semantic system, let's say, so, but it has the capacity to implement contingent mappings.
Okay.
And okay, yeah.
So it would mean that actually the last point here, the goal-directed systems, instead of using semiosis, actually they are generating them.
No, they are actually then using it
Because there's no semiosis which is independent, as you're saying, is a possibility, but actually what generates does is the fact that you have a goal-directed system that exploits.
I would say it like this. Yes.
Is it correct?
I would say, yeah. So that would be the next step in a way, yeah. So first you have the capacity to do it, the capacity to make the contingency. And then of course the question is it used or not? So if it's never used, or is it used? So in order to make a semantic system, of course it has to be used.
Okay.
Well, in cybernetics, the simplest semantics is from perception to action. That means you sense some perturbation, and then you map it onto the right action to deal with that perturbation. So if you sense that the temperature is too high, you map it to an action that reduces the temperature. If the temperature is too low, you map it to an action.
Okay, so can I? Yes.
But in these cases, it's almost trivial. But when there are many options, then indeed it becomes interesting to have some kind of a system that mediates between the two and that can learn different mappings. And to be able to learn different mappings, there must be a contingency. If the mapping is hardwired, you can't learn.
But let's take this example: temperature and reaction to the temperature. So there are two ways. There's a contingent reaction and non-contingent. So the non-contingent would be a protein. Yeah, you heat the protein and then you get the egg, it becomes sticky. Yeah. So this relation you cannot change. I mean, that it becomes, I mean, that's a direct physical reaction of the protein that changes its conformation. Yeah. But if you have now a temperature sensor and then the temperature sensor hot, and that you can change. So that means that in the basic cybernetic definition, there's no notion about what kind of physical process you are underlying assuming. So you don't have the alternative thing that is there.
Well, no, but you could have a negative feedback that is natural. Like one slide I didn't show was a feedback between grass and rabbits. More grass means more rabbits means less grass. So there will be this parallel limit cycle, so that there's a negative feedback.
I agree.
And here there is no contingent mapping,
but
it's a negative feedback. And in this case, it will not go to a fixed equilibrium, but you can imagine ones that go to a fixed equilibrium. So they are like the thermostat, but they're not contingent.
I fully agree. Yeah. So when you have, let's say, even when you have a reaction A reacts to B and B reacts to A, then the A is inhibiting itself. So when you look at the differential equation, you have a negative feedback. So I don't say that I'm talking about feedback controls. That is why I use the German word Regelkreis, and I don't mean negative feedback. Because negative feedback, of course,
a modulated negative feedback, where there's something that needs to interpret the sensation or the perception and map it onto the action, where there is no immediate connection from
exactly. And that is, let's say, true in all these feedback control circuits that I think thought about control theory. And of course, the other one, maybe we can call emergent control, where the negative feedback is some kind of emergent process of all the small interacting entities that generate an.
And there we get into the interesting question of the origin of goal-directedness. At the lowest level, goal-directedness would be just a system finds its feedback just because of immediate mappings. But then if it needs to adapt to more complex circumstances, it means a larger variety of disturbances, then it becomes handy if it can learn to experiment with different mappings. And then an element of contingency may facilitate this learning, may enable this learning. And that may actually be the role of the genetic code as a way to enable learning lots of different reactions to different
Of course, that's right. Yes. Good.
More questions?
Of course, we have more discussions later.
Yeah. we go to the coffee break. I'll bring this page so we can quickly, which is.
Leave this.
Peter, do you want the coffee?
No.
No? Okay. So, will be served in a couple of minutes. So
should I?
And this is going to be 15 minutes.
I have this kind of